John White was a key player in two of the LMB's biggest achievements, the complete reconstruction of the nervous system of the nematode worm C. elegans and the development of the confocal microscope. He started his scientific working life as an electronics technician at the MRC National Institute for Medical Research in Mill Hill, where he did a sandwich degree in physics, moving over to the LMB in 1969 to work with Sydney Brenner. His interest and aptitude for developing innovative solutions to difficult technical problems have driven his career ever since, both at the LMB and subsequently at the University of Wisconsin-Madison, where he moved in 1993, retiring in 2008. John is a Fellow of the Royal Society and, together with Brad Amos, Richard Durbin and Mick Fordham, won the Queen's Award for Industry in 1990 for the development and production of the first confocal beam scanning microscope. John, thank you very much for talking to us today. I'd like to start by asking you about your move from NIMR to the LMB. How did that come about?
Well, it was a strange occurrence, really, because I had just finished my degree, a physics degree, and I had this wonderful sandwich course, which is you don't hear much about these days, but it was a really effective way of enabling someone like me to actually get a degree whilst working, and so I could pay for my upkeep. But anyway, I'd got interested in computers there and done some work displaying three-dimensional models of molecules and being able to animate these and rotate them around. So we had a computer that was capable of doing this. And I was quite interested in progressing with this sort of work. And there didn't seem to be a whole lot of opportunity by the time I'd graduated. And so I looked around and got a job offered designing computer-controlled telephone exchanges and was really thinking about taking up the offer. But anyway, the head of the department at the time heard about the fact that I was thinking about moving on, and he came and spoke to me about this project that was going on in Cambridge. And I remember him saying there's some bloke there that's interested in worms. And he's interested in doing computer reconstructions. Why don't you go and talk to him? Mm-hmm. And so I was intrigued, but I think more out of a sort of polite duty to the MRC, who'd really done very well by me in terms of funding my education and giving me insight to biology. I trotted along to Cambridge and put on my best suit and went and met Sydney Brenner. And it was really quite extraordinary meeting, because the whole atmosphere at the Laboratory of Molecular Biology was so totally different from the NIMR. There's a bustle and energy which one could feel, and the corridors were full of humming equipment and centrifuges and incubators and this sort of thing. And then I was taken into a room and met this extraordinary fellow. This looked a little bit like a Neanderthal man who was chain smoking, and he was Sydney Brenner. And I also met Tony Stratton there, and he started talking 19 to the dozen about his ideas about using nematode and being able to reconstruct nervous systems and using computer graphics to do it, and would I be interested in this? And I was somewhat overwhelmed by all this, and I remember sort of going home from that interview with my head spinning, and I thought Sydney was a little mad, but I thought it could all be quite fun. So anyway, I was becoming sort of favourably disposed to the idea. And then a few days later, Sydney called me up and said, well, do you want to come? You'd be a transfer to a different MRC unit, so there'd be very little paperwork to do. And that was it. I was hooked. And so I duly went there a few weeks later and started this new phase of my career.
Mm-hmm. So this was right at the beginning of the worm project, I guess.
Yes, it was. I think Sydney really started getting it in about 1965, and this was in 1969, where he had done a lot of the spade work, and he had basically worked out ways of culturing it and worked out ways of getting mutations and ways of mapping mutations, and he got some quite interesting mutations of behaviour. And he also had a wonderful technician called Nicol Thompson, who was an extraordinarily gifted electron microscopist, and he'd started cutting into different worms, looking at various structures, and Sydney started to piece together the structures in the head, the sensory apparatus and the feeding apparatus, and started to, I think, realise that in fact you could piece this together from serial sections. And so he had done some drawings on transparencies using Chinagraph pencils, and you can see these sort of reconstructions coming together. And anyway, he was full of enthusiasm for getting the latest in computer technology. But this was in 1969. The latest in computer technology was pretty lame compared to what we're used to now, but it seemed maybe marginally doable. But I was quite fired up by all this, and so that's what happened. And then when I started, of course, we hadn't ordered the computer or anything. We just were deciding what we wanted, and the thing that Sydney had me doing was learn to pick worms and isolate mutants, which is what I did for a bit. And
so was this your first exposure to biology then, or had you done stuff like that before?
Yes, first hands-on biology, because I done a little bit in the NIMR with electrophysiologists designing a sort of patch clamp apparatus and this sort of thing, but it was the first time I'd actually sort of handled the meat, so to speak.
So how was that then?
I was very excited because this small thing, really tiny thing, was really quite a rich repertoire of behaviour that one could see under the microscope, the way it wiggled around and located food and all this sort of thing. And then I learnt from the other people there, like Tony Stratton, that it only had about 300 nerve cells. And so this became extremely fascinating to me because, I was an engineer and an electronic engineer, sort of computer scientist, and it became apparent that what we could possibly do with this is get the circuit diagram of the nervous system of an animal, the first one. And so this was an exciting prospect. So that's why I really got into this. But anyway, when the computer came, it was sort of interesting as well because it was by no means plug and play like modern computers. It was had a very primitive software, and it had disk storage, which was innovative at the time, but it had no software to actually store things on the disk. And so I realized that I was going to have to write what essentially was a disk operating system for this computer before we could start to do anything. So that's what I did. But then the other interesting part of this is the other person that got really into this was Sydney Brenner. He was dying to get his hands on computers. And he was been thinking for a long time about computers. And I think he had a friend in South Africa who was turned out to be an early computer pioneer, but I think he sort of fired Sydney's interest up in computers. And so, and I think the thing that really cemented his interest is when Sydney Brenner saw Francis Crick and Jim Watson's model of DNA. And because what he realized was this is information. And at the time, there was a very famous computer scientist, John von Neumann, who was speculating on how organisms can reproduce themselves. But this is without knowing the structure of DNA, of course. And he couched his ideas in terms of information. And in fact, they must have a mechanism for basically reproducing the information and constructing the system which reproduces the information. And this is the sort of formal description, if you like, of living organisms. And so Sydney was very primed, to do this, and he turned out to be a very competent, a very gifted programmer, because at that time we weren't using all these fancy high-powered languages like Perl or whatever. We were using pretty low assembly language, where you were talking in the basic instructions that the computer hardware did. But he seemed to relish in this, and he developed a bit of software called Track, which had been developed elsewhere, but it was a piece of software which basically can analyze strings of characters, so it would be called scripting software now. This is all part of computers, but it was very useful for developing the system, because then we could write simple instructions about how to do things like save files to disk or search for files on disk, using this scripting system. So it was an important part of the system that we were developing. But then the other interesting thing that happened at that time, by this time we were up to about 1971, 72, is that Sydney's started to get very interested in gene cloning. And this was at the time when restriction enzymes were being wiped out, and various other people were suggesting and even demonstrating that it's possible to take bits of DNA and clone them. And Sydney got really fired up about this, and he carried his interest in computing and started changing his focus towards gene cloning, and essentially dropped the anatomical project. So he played no further part in that. But as luck would have it, I inherited a very gifted technician called Eileen Southgate, who'd worked with Tony Stratton, who'd done a bit of anatomical reconstruction on a bigger version of the worm, and had now relocated to Madison. And so she started working with me, and of course we had the superb ability of Nigel Thompson and his electron microscopy. And so just about the time that the computer system was coming to completion, or just before that, I could really start to get involved in the reconstructions. Quite seriously.
So was it typical of Sydney that he got very enthusiastic about something and then dropped it and then moved on to something else? Or?
Yes, Sydney's the sort of person that he was essentially serially monogamous in terms of his sort of scientific. Yes, and this is very Sydney-like behaviour. Yeah, he liked to initiate things, and start things. He liked to light the fuse, and stand back when the fireworks started to burn.
So was there an overt decision to do the whole worm, or did it just evolve into that project?
Well, Sydney was, I think, a bit coy with me, first of all, and I think maybe because he didn't want to put me off. And so we were talking about ganglions and the ventral cord. He was quite excited when we got some stuff out the ventral cord and found some regularities. But I just remember that we never talked about him, the whole system. But I remember one day he was showing someone around, as he often did, and then just as he was. That they were leaving in sort of sotto voce, he said, and soon we'll be doing the whole nervous system. And I sort of started taking it back with this.
So you think that was always in his head then? He just didn't dare tell
absolutely, you? Yes.
So this was 1991. So you'd switched onto
71.
You'd switched onto a PhD track at that point.
Yes.
So what was that a common thing for people to be doing in those days, to move on to doing a PhD from? I guess you started as a research assistant.
Yes, I was, and it was unusual. I didn't apply to be a graduate student and this sort of thing, But yeah, it was more informal occasion, so it seemed an obvious thing to do. And the nice thing about it was that I could essentially, my PhD was in two parts. Half of it was the development of the computer reconstruction system, and the other half was the nervous system, or the ventral cord, of the part of the nervous system we'd done up to that time. And so essentially it was relatively easy for me to do, and it was good to, as a good precursor to sort of writing the papers.
Were you surprised to find yourself in this situation, given that you'd not bothered to go to university and you'd not wanted to do this stuff?
Yes, a bit bemused by it. Yes. Because a lot of people were very career oriented, and I think I was the only grad student at the time at the LMB that had not gone to public school, and most people had careers that they wanted to do, but I just enjoyed messing around. I think the thing that's always driven me, the most powerful motivating factor, was curiosity, really, and I found these things really did sort of satiate my curiosity quite a lot. I found them absolutely fascinating, and also the wonderful freedom to be able to pursue a project as you want it and as you think it ought to be done, and having extremely competent people around you like Eileen Southgate and Nicole Thompson. There's really quite a heady atmosphere, really, I think. Yeah. And also, of course, the culture of the lab was extraordinary, because we used to have this coffee room, which we met every morning for coffee, and Sydney would go on about various things. But for me, who'd come from a sort of physics engineering background and knew very little biology, let alone chemistry, it was a very good way for me to learn, just by osmosis, so to speak, being constantly bombarded by all this chatter about DNA, RNA, and biochemistry, and this sort of thing. It really sort of filled in many of the gaps in my background knowledge, which was very useful.
I mean, it's interesting what you said about recognising that you could make a wiring diagram. I think, maybe that wouldn't have been so obvious to someone who'd been trained as a biologist at that point.
Well, I think so, yes. I think they wouldn't have thought of nervous systems in that sense, and of course, the other interesting thing for me was the developmental aspects of it. It was, this wiring diagram wasn't made with someone with a bit of wire and a soldering iron. It developed, evolved over time, and it was formed as part of the developmental processes.
Yeah. So just going back to the atmosphere here at that point, was there any sort of hierarchy?
No. That was a lovely thing about it. In the MRC in Mill Hill, there was separate coffee rooms for the technicians and the scientific staff, whereas here, everyone was mixed together. The canteen was a great centre for mixing and discussion, and people like Francis Crick, it didn't matter who you were. If you were interested in something that he was interested in or was thinking about, and you said something or a suggestion or something like that, he would listen to you. He wouldn't dismiss it. So there was this wonderful interchange and very lively conversations. And it was also extraordinary because it was the first place I'd worked at where there was a large presence of American people working there, and I was aware of the sort of cultural differences, and I think there was an interesting synergy between the two cultures, because the Americans were very gung ho. the American postdocs we get were extraordinarily high quality. They were highly motivated because they didn't get any money from the MRC. They had to come on their own money. They had to get their own funding. And, they really wanted to come over, and this is the place where DNA was discovered. They wanted to be wound up and pointed in the right direction and go. But they found that us Brits, often like to go up and have our cup of tea and really talk about things. And, often they didn't seem terribly relevant to what they were doing at the time. But there was a lot of toing and froing of information, ideas in a very sort of relaxed, low-key sort of environment, which I think was highly productive. It wasn't meant to be, it wasn't designed to be rich. It just happened. Ideas were swapped around and people didn't feel intimidated about saying silly ideas, because they would all be discussed. And so I think it was very fertile at the time. And also, the other extraordinary thing about working with Sydney Brenner, particularly, when we got onto the reconstruction process, as I say, he was off in another direction. Whilst I was doing my PhD, he was a very strange supervisor. He never asked me what I was doing. I wrote up my thesis and bound it. And I said, "Oh, Sydney, I'm going to submit my thesis now." He said, "Well, I better have a look at it." He looked at it then. But it was lovely to have that freedom, I think. And it was obviously implied, I think, a sense of trust from him. The fact that he didn't meddle or, want to see you try to micromanage and this sort of thing meant implicitly to me that he trusted us to sort of, forge our own path.
Do you think the worm project wasn't universally regarded as a good idea at that point outside the LMB. So were you insulated from that within the lab, or did people realise it was a bit of a risk, especially
we were insulated, particularly the cell biology. Even within the lab, I think there were factions that thought that this was, a bit of an indulgence for Sydney Brenner, and they didn't take it too seriously. And he really did an amazing job of getting the project established around the world. He had wonderful charisma. His talks were hilarious and but penetrating. He had extraordinary wit. Yeah. And he went around the world and gave talks and basically attracted. That's how these very high-powered, very able postdocs came. He attracted them.
Yeah, I think you described him somewhere as the most intellectually seductive person you'd ever encountered, which I thought was...
Yes, well, that's what he did. Yeah. And the way I described him at that time, he was like the Pied Piper of Hamelin on speed. And but yeah, it was quite extraordinary. But then, as I say, he drew these people in which started working and doing stuff together. But he didn't lead it in a sense. He was quite happy for people to get off and do their things. So if you wanted to speak to him about what you were doing or something like that, or it was related to his kind of interest, he would be delighted to do that. It's not so we were shut out or anything, but it was this sort of sense of freedom and free exchange of information that was really quite a heady atmosphere, I think. Yeah. It was quite unlike anything I've known since in the academic world.
So let's move on to the mind of a worm then. So 1971, there's you, Nicol Thompson and Eileen Southgate. Yes. Nicol is salami slicing worms and making electron micrographs of them. And what were you and Eileen up to?
Well, Eileen was printing these out, and she was sticking them all together in montages. So we would take a whole section of a worm, and to get the sort of detail that we want, we needed 6 prints. So these were stuck together by sticky tape and then trimmed, what have you. And then we made a big discovery, a sort of enabling technology. There were these things called rote ring pens that were used by draftsmen, and you could label quite fine nibs. You can label individual neurons, and they were coloured and what have you. And but also critically, we could erase the label with a Q-tip dipped in alcohol, and so that was essentially how it was done. And then sort of maps were made of where things were, and there were sort of silly sort of little innovations that we had to do. Like one of the technical problems with EM at that time is often the sections are quite compressed, and it varies from section to section. And so if we wanted to plot where a synapse was, this was difficult because the next section it may be squashed, and so any coordinate system you had would give you the wrong answer. So what we did was make a set of transparencies which had different degrees of squash in them, and had scales on them. And so with any section, we could find the right transparency that had the right degree of squash, and then Eileen can measure the position of the various synapses and what have you, and then when you plot these out, then you don't get the squashing effect. So. And
these aren't, I mean, they're not very easy to look at, are they? No. It's a horrendous job.
Well, Eileen was quite extraordinary because, she had lots of abilities that I never had. And I found out quite late in our relationship that her hobby was, and what she used to go home to do, was putting 1,000-piece jigsaw puzzles together, which was really quite extraordinary. She had a wonderful visual memory, and unlike me, she was very meticulous and kept excellent notes, and basically very methodical, and would just keep going, and it was quite wonderful, really.
It must have taken the most extraordinary patience to do this as well. Yeah. So the two of you were doing this?
Yes, and then how it worked was, typically about 10 o'clock in the morning I would go and see her, and she would talk about her and what she'd done and any problems, and there were lots of problems, because there were gaps in the electron micrographs and all this sort of thing, and trying to what to do, and whether this was a synapse or whether it wasn't a synapse, and what was the criterion for deciding whether it was a synapse or a gap junction. And then, so this would go on, and then I would leave her, and then she, would carry on and do things until she developed another list of things that we ought to make decisions on. That's how it worked. And it's a very, very nice collaboration. So the progress seemed glacial.
So you were doing other things at the same time as this, were you?
Yes. The other thing I got involved because simultaneously with the nervous system reconstruction, there's another extraordinary thing happening with C. elegans, and that is that John Sawson, who was my colleague and lab mate, we worked together, was working out the cell lineage of it, and he first of all did the cell lineage of the adult and the larval stages. All sorts of interesting things were discovered, the way that the nervous system developed, and he also eventually went on to reconstructing the embryo, which was a complete tour de force. It was all done just by eyeball and good microscopy, and it was just wonderful. And so this is happening at the time that we were putting the nervous system together. So it's very exciting because, we were seeing these patterns, regular patterns, which we could ascribe to certain types of neuron and give them class names. And we could see from John's work the lineage of these neurons. And so we could tie the two together. And so this, I think, was extremely interesting. At the time we were doing the ventral cord, when we got into doing the embryo, or when John got into doing the embryo, I should say, we had to tie up the things that he saw in the embryo with what we knew from the adult structure, and that was quite a challenge. And Nicol Thompson did this heroic work because he had to take a single embryo which had been lineaged by John Sawston. It requires an incredible amount of effort, days of almost constant looking down the microscope and mapping out cells. And then he had to prepare this one embryo, this tiny embryo, and fix it and section it for the electron microscope. So all sorts of things that had gone wrong. And eventually it turned out to be not terribly good one. The quality was poor, but it was good enough. And so we were able, all three of us getting together, that's Eileen, John and I, is to work out what all the cells were in the embryo and find out what, so we could then take the identifications of the cells which we'd made from the adult reconstruction and take them right back to the embryo so that we knew how all the cells were born. We knew their lineage in that sense. And so the lineage by itself was considerably enhanced by the knowledge of what cells came at the terminal twigs of the lineage. So that was an extremely exciting period, and then a couple of things happened there, because as soon as we started to look at the embryo, the lineage was interesting, and it wasn't totally symmetrical. It wasn't just a tree-like expansion of the original fertilized zygote. There were asymmetries in the lineage, and it was very interesting to try to. There was lots of discussion about what was going on here, and there were various people that thought that there must be some intrinsic program which is basically specifying the lineage. And there's another school of thought which I think I was more in tune with, which said that basically these asymmetries were maybe based by communication between cells. Anyway, this was a big discussion point, and I wondered about this, and I thought, well, wouldn't it be nice to be able to kill or remove a cell, a particular part of the lineage, and seeing what the other cells did? There had been reports of people being able to do cell killing using a laser, a flash laser. It was a dye laser, which is pumped by a flash tube, and was able to get sufficient precision to kill cells without causing extensive collateral damage. And so, given that what expertise I had was really more in the sort of physics engineering side, I got into the workshop and tried to design a system for killing individual cells. And there's a wonderful workshop at the LMB. And anyway, we did this, cut a long story short, and basically we could kill individual cells. And John Sawston got quite good at this because of his familiarity with the lineage. And then other postdocs, we had a very gifted postdoc, Marty Chalfie, who was interested in touch response and this sort of thing, and he identified various neurons which might be involved in that. And we were able to do experiments where we killed particular cells and see what would happen to the development of the system. It was extremely useful because it gave us quite deep insights to how things work, and it became even more important later on when people started looking at mutants of development and this type of thing. And there were a whole class of mutants which were isolated, which had bizarre phenotypes. It's either affect one thing here and another thing there, and no one could really make any sense of it. And then it was discovered that if you killed a certain cell, you could replicate these defects, and basically what the defect was a failure of communication between the cells, and other people picked this up, like Judith Kimball came over and picked this story up, and it was an extremely interesting story which came to really discover a whole lot of signaling pathways. I wasn't involved in this directly, but it is, I think, a very important part of the nematode story. That they did some very pioneering work in opening up these signaling pathways between cells which determine cell fate. And
so it's a sort of interesting mix of, on the one hand, you were doing a project that was about as untechy as you can get with a rotary pencil and some pictures, and then on the other hand, you were doing this sort of extremely complicated technological development. So, well,
yes, most of the heavy lifting with the neuroanatomy reconstruction was done by Eileen, because she was the one that sat down all day and put all the numbers on the papers there, and basically we got together to discuss various things and what it meant, and try to put the whole thing together. But yes, I think one tries to do things. You have got sort of training or capabilities or interest in certain skills. There are problems to be solved. One tends to use the skills that are at one's disposal to see if you can think of a solution using those skills. And,
so The Mind of the Worm was published in 1986, I think. So 340 pages long, 13 years of work.
Well, it was actually submitted in 1984. It had a long, it was a long gestation period.
Yeah, so I wrote some numbers down about this project. So the 302 neurons in the adult hermaphrodite, 5,000 synapses, 2,000 neuromuscular junctions, and 600 gap junctions.
Yes, about that.
and I gather that Eileen only made one error in the whole thing.
Well,
or maybe that was you.
Yeah, probably maybe. But there was one cell misassignment, which I think was picked up.
I mean, that is extraordinary.
Yes, well, I think it really reflects on the meticulousness and dedication of Eileen. And
did you ever worry that she was going to leave and that you'd have to end up doing it?
Not really, no. It was interesting that she got into it because I think, as I said, she liked doing jigsaw puzzles, and she saw it as a puzzle. She wasn't interested in how the nervous system worked or how it developed as such, but she saw it as some visual structure. And when she got more familiar with it, she realised that there were lots of these cells with processes which interacted with each other and connected with each other, and so she was driven on, by her own sort of fascination with structure, and so to her it was a big challenge, and so she may well have left. I'm sure she would have been very employable by many groups, but I think she really liked doing it, and she felt, although progress was very slow and incremental, that she was putting it together. And so
I guess if you do jigsaw puzzles, you don't like to leave them unfinished as well, do you?
That's a good point. Yes.
So once that was finished, you moved on to other things, I guess. So at that point, were you running your own lab?
You see, I think that gives the wrong impression. We weren't really running labs as such, and it was, they didn't really have the whole concept of labs that they do now. You did have graduate students and postdocs, but they intermingled quite a lot, and many or several of them applied to be a postdoc, say with Sydney, and they would end up by working with John Sawston or me or other people, and so we were their de facto sort of mentors, but it wasn't really that sort of rigid structure. I think it's changed now, and it certainly was very different from the American sort of structure. There was no territory we had rooms where we worked, and there was a room for the electron microscope and a room where we could put the optics and this sort of thing. And various postdocs came. You wondered where to put them, and you do a bit of horse trading, try to find a bench and this, that, and the other. And basically, if you want to write up anything, you went to the library. And then you had to be sure that when you finished doing that, you took all your papers away as well, so someone else could use it. So, I think Sydney and maybe even Francis, though it wasn't deliberate, thought that the overcrowding was perhaps not a bad thing. And the other thing, the effect it had is that quite a few people went essentially on the night shift. Partly out of choice because they were nocturnal anyway, but partly if they didn't really like working in crowded conditions, they would work overnight. So the lab was sort of open and running 24 hours a day.
So I just want to move on to the development of the confocal.
Well, that came about again at the time of the cell lineages that John was coming out with, because we were interested in this question of the asymmetries in the lineage, what have you, and I really wanted to see what was happening at the cytoskeletal architecture, during the various processes of asymmetric divisions and all this type of thing, and it all seemed a bit difficult, but then. Two sort of developments outside our lab sort of piqued my interest, and one was the development of the technique of immunofluorescence, whereby you could stain things, stain structures of the cytoskeleton and visualize them. And this was a tremendous breakthrough, to be able to stain a cell and look at all the microtubules or the actin filaments or the intermediate filaments or whatever. It was a tremendous breakthrough. So that was exciting. And so I was interested into trying to see what happened to the cytoskeleton during these cell divisions. And I had a very gifted grad student came to join me, Tony Hyman, who was interested in it and working on this, because there's all sorts of interesting things that happen during development, interesting flows of stuff around the cortex, but we couldn't really see it. And the reason we couldn't see it is that the embryos are fairly thick and round, and where they published all these wonderful pictures in the front cover of Cell or something like that, these were very flattened cells which had leading lamella, and you could see all the architecture there because it was very squash flat. But to try to see this inside a solid embryo, it's impossible. You've got this blur, and the reason was the optics can focus on just one plane. Because it's fluorescence, you're getting light emitted from above and below, and it just gives this flare, this out of focus flare. And anyway, I was pondering this, and I remember when Tony, first tried these things, I was disappointed in the results because we couldn't see anything. And then Aaron Klug gave me a paper to review. He was obviously interested in instrumentation. He gave me a paper. It was about confocal microscopy. It was from a guy called Brackenhoff in Holland. And in fact, he used confocal microscopy. I can't remember what his sample was, but it was a biological sample, and showed that you could remove this out of focus interference. And the sort of microscope that he used at that time was really totally unsuitable for our particular application. It was a great thing. It scanned lines across the sample, but it did so by having this huge thing which physically moved across it. And it was very slow, and it was very sensitive to vibration. It made a very good seismometer. And so it wasn't suitable. But I wondered whether instead of actually scanning the microscope, we could scan the light beam using mirrors that could be deflected. And, there was technology for having mirrors that could be driven by actuators and sort of thing. So we thought of trying to do that. Now, there were other attempts at making a confocal microscope using spinning discs and what have you. So it was clear that the technology was possible and appropriate. But you really wanted a way that we could use a normal microscope and basically have something that would augment what we were seeing in a conventional fluorescence microscope. We wanted to go beyond the sort of proof of concept. So we really wanted to get results out of this and to find out about. So it was really problem driven. And I thought, well, we could make a system that scanned the beam using these mirrors being driven by electronic actuators, and it'd be possible to make something that we could put on top of a conventional microscope, which would convert it into a scanning microscope.
So had anybody thought about doing something like this before for microscope?
I didn't know anyone had, but the confocal microscope. Scanning laser microscopes had been thought of, but not confocal ones. Anyway, it got to go down to the workshop, and they have a wonderful workshop with an amazing bunch of guys.
Because I guess you couldn't even think about doing these things without knowing that you had that workshop to support.
Absolutely, no. And I sort of sketched some ideas and what have you, and started talking to them, and they were extremely enthusiastic about this.
So how does that process work then? You go down and you have a chat and.
Well, I drew a few things out on a paper and said, we would like to do this, to make this microscope. And I found various devices that we could use. Initially, we didn't use mirrors that were deflected by electronic actuators. We used mirrors which were rather like these things you see in discos. They're multifaceted, and they swept round. And that was the first one that we made. And I'd go down there and see them. And there was one guy down there that was really excited about the project. This is Mick Fordham, very talented engineer, and came up with all sorts of ideas. And anyway, we talked about this, and I think, well, we might be able to knock something up. And it wasn't that easy because we needed things like lasers, and we also needed quite a lot of fancy electronics. But we managed to improvise, and they found a laser surplus to requirements, and the electronics to do all the scanning. And I found I had an oscilloscope and managed to use the time generators, the scanning generators out of that, and take wires out of it. After much fiddling around, we found that it worked, and it was hugely exciting. And basically, what we were able to do is look at a nematode embryo, look at one that had been stained with antibodies to microtubules, or the microtubule protein, and compare it with the images that we get on a fluorescence microscope. And
so this
is what we have here.
That's extraordinary.
So this is the same image. This is a nematode embryo. Here are the two cells, and these are the centrosomes, things called centrosomes, which are a very important part of cell division. This is exactly the same specimen looked at with the fluorescence microscope, and you can see very little detail in it. Whereas with the confocal, and this is this pile of junk that was just on a bench. You can see the sort of little structures of the microtubules, and so this was very exciting.
You must have had a queue halfway around the block when people found out that you could do that.
Well, yes, lots of people got very interested in it. So anyway, having got this result from what we called the Mark I, we decided to make a slightly more packaged version of this, a more reliable version of it. So, but using the same technology as was in the Mark I, it was put in a box and tried to make it a little bit more reliable, a little bit more stable. And this became known as the Mark II. And then people did start to get interested in it. And coincidentally, at the time, one of the researchers in the lab was Sean Munro and Hugh Pelham were doing some very exciting work on protein trafficking in cells. And basically, they found out the mechanism by which proteins traffic to these structures called the endoplasmic reticulum inside cells using a little sequence, a little message sequence called KDEL. And they discovered this. This was a very significant discovery. But the trouble was, they couldn't really see what was happening because the ER is a very dispersed membranous organelle within the cell. And when they looked at these cells, even though they were fairly flat, it just looked like some haze. And so somehow they heard about it and they brought some samples down to the Mark II and started actually seeing details of the ER. So this work was published. I can't remember. I think it must have been 1986. The picture made the front cover of Cell. But at the time, this was still fairly early time in the development cycle of the confocal. We didn't really want to talk about it in that sense. And so there was just rather a coy mention of the fact that we use a confocal microscope, which will be described later, sort of thing. But yeah, so it was clear that it had potential. And at that time, there were serious discussions about maybe we should commercialize this. And I think Aaron Klug was very enthusiastic about this. And he helped us, found some sort of hidden pots of money in the LMB, which enabled us to buy a thing called a frame saw, which we could store images on, rather than have to take photographs of the scanning spot across the oscilloscope screen and various other things like that. And we wondered about how to commercialize it. And I didn't want to basically, devote my whole time to developing the confocal microscope because I was really interested in this whole project of C. elegans and its nervous system and the development and what have you. But anyways, to cut a long story short, we did meet a company that had been involved in making some of the early versions of the scanning stage microscope. This is where the whole thing moves across, and they were interested in this beam scanning, and they agreed to commercialize it, and they but they were interested in us producing a sort of pre-production prototype, which we did, and we changed the technology, so we actually went to using these mirrors I was talking about, which are deflected by these little electromagnetic actuators, and tried to make it compact and tried to make it into a sort of instrument that a lab could get and put on their microscope and plug into their computer and work. And then the Bio-Rad started. I think they made the first batch of 5, and then obviously I had to help them to get these 5 things going. And then I had quite an exciting time where they sold these very quickly.
I bet they did.
The thing it was interesting actually because it's one of the few things, new bits of technology, which you can immediately see the advantage of, and they put this prototype that we had, I think, in some of the exhibitions. I think it might have been in the ASCB, the American Society for Cell Biology meetings, and showed it. And you can immediately see, you show them a sample, a typical sample that people have been used to, and then switch it in, and most people say, "Oh my God!" And then so, there was a huge amount of interest in that because it was very apparent that it enabled you to do things that you couldn't do otherwise. And given that immunofluorescence was becoming a very established technique at that time, lots of people started putting these in their grants, so much so that the NIH started to get a bit alarmed about this because they wondered, is this just the latest craze or is this real useful technology? And so I think they put a moratorium on it for a time. And there was even a cartoon in journal Science, a very high-profile scientific journal, with a picture of Santa Claus, and a little girl going up and Santa Claus said, "Do you really want a confocal?" and so. Yes, and it was sort of interesting working with the commercial company. You realise it's just so different from working in the laboratory. And I remember once them saying to me after drinks after some sort of display, they typed up and saying, "Oh, this is very exciting technology." And after a while, I realised it was very exciting technology for different reasons. From their point of view, it was exciting technology because they could sell things. For me, it was exciting technology because we could do things which couldn't be done in other ways.
Gosh, I wonder how many confocals there are in the world now.
There are quite a few. It's fairly established.
Yeah. Hundreds of thousands.
I don't know. But it was interesting. And it was interesting to me just to sort of dip a toe into this commercial world and see what's involved in developing something like that. And there are many ways of doing it. And the way we did it was rather unusual. And actually, we made the prototype, which was very similar to what was ultimately sold.
So you were not tempted into industry in any way?
Not really, because I was really sort of hooked on this
on the worm.
Yes, this project that we were working on, to me, although my skills were in instrumentation and that sort of thing, it was means to an end. It wasn't, I didn't really want to become an instrumentation engineer.
So, out of all these many amazing things you've done in your long career, have you got any that you're particularly proud of?
One of the things that I really enjoyed doing, and I don't even know whether it's how correct it is, but it was sort of interesting. It was somewhat different, and it did actually use the computer for the reconstruction project. I got interested in the process of cytokinesis and worked out a way of modelling it. And we had this theory of how cytokinesis might work, and we managed to put this into a computer model and then made an animation of a dividing cell. The computer, of course, it's easy to do now with YouTube and all these graphics, but what we had to do was we had to do one iteration, and then we had this camera, old-fashioned film camera, which we could take one frame at a time and then inch it on and do another frame. But yes, we could get some reasonable, and I found that most satisfying. I think we had various ideas of what was going on there. I think some of them were correct, and some of them, maybe not so much. But I found that quite exhilarating, to be able to, think of how something works, and then be able to do experiments by modelling and this sort of thing to see whether it was feasible. And the modelling was quite informative, because although you can make a model which can mimic why something works, it doesn't really mean to say that you've got it right. There could be many ways of skinning a cat. But if you can't do it, it can show you when something is wrong and the approach is wrong and it's insufficient. You need something else, and it did that, so that was, I think, something that gave me most pleasure. But it's I'm certainly not known for that. It was not a major publication in terms of my output. But,
you can still have favourites that aren't famous. Yeah. John White, thank you so much for coming to talk to us.
Okay, I enjoyed it. Thank you.